I'm actually going to do really cool work in weird microbes, and that's all I'm going to say, and then you can.
Great. Awesome. I love it. I get more time to talk. Yeah, so I'm really broadly interested in thinking about how the evolution of the actin and microtubule cytoskeletons can help give rise to eukaryotic diversity. And so that's a big umbrella, but today I'm going to tell you about one organism that we work with in my lab and sort of focusing on its developmental biology a little bit. And so these are all images of types of fungus called chytrid fungi. And so those are going to be what we're talking about today. And really, fundamentally, the development of cell shape is really important for the physiology and symbiosis that fungi can sort of do. And that's similar to most organisms. But I think among fungi, there's some really clear examples of this. So one example is this concept of dimorphism. So fungi can adopt multiple forms, and people have thought about this when thinking about pathogenic fungi. And so this transition between these different, like a yeast-like form or yeast form and an elongated form is really important for the biology of these organisms as pathogens. This is true for plant pathogens as well. So some plant pathogens can make a structure called the aprosorium. This is a structure that fungi can use that helps them increase their internal pressure. And so they can literally punch through the cell wall of plants to gain access. And so the development of cell shape is not important only for the bad stuff that fungi can do, but also the good stuff they bring. So for example, this is a structure called an arbuscule. These are structures assembled by mycorrhizal fungi. These are fungi that form a really important symbiosis with plants. And in this case, in these types of arbuscular mycorrhizal fungi, these structures are built by the fungi inside the plant cell walls. And the frilly nature of this structure increases the surface area in which the fungus and the plant can interact. And so these are just a couple examples of how control of cell shape in fungi is really important for their physiology and success. And so this leads to a really fundamental question, which is how do cells control their shape? And obviously the answer to this depends on what kind of cell you're talking about. So animal cells, for example, control their cell shape from the inside using networks called the actin cortex. So this is a layer of actin that lies underneath the plasma membrane, and the plasma membrane lies on top. So animal cells can change their shape by changing the organization of these actin networks. And that's fundamentally different from how fungi do it. So fungi are encased in a cell wall, and so they control their cell shape by controlling the structure and the shape of the cell wall. And really, it's a combination of two activities that helps fungi control their shape. The first is polarized growth of the cell wall, and second is inflation of the cell wall using internal pressure called turgor pressure. You can think about this a little bit like a mylar balloon. So the shape of a mylar balloon is controlled both by the shape of the plastic as well as the degree to which you inflate it. So the question we'll be talking about today is, where did this system come from? And I think this is a really interesting evolutionary question because sister lineages of fungi lack cell walls. So fungi are shown here in purple and blue. They're hugely diverse groups of organisms. And their sister group are the animals, okay? And so this together make up a group of eukaryotes called the opisthocons. And we've already talked about animals controlling their cell shape from the inside using actin. And it turns out that organisms that lie outside of the opisthocons do the same thing. So this includes amoebozoans, different types of amoebae, including Dictyostelium, which control their cell shape from the inside using actin. Okay. So really, the fungal lineage during its early evolution was able to move away from using actin on the inside to control its cell shape and really control its cell shape using these cell walls and turgor pressure. But it turns out that most of what we know about the fungal cell wall comes from studying a single group of fungi called the dikarya. And so the dikarya are shown here in blue, and these include all the famous fungi. Okay, so all the mushrooms, the yeasts, filamentous fungi are all examples of dikaryotic fungi. And even in dikarya, it turns out that cell wall composition varies across species as well as across developmental time. So for example, Candida albicans has a cell wall, which you can see via TEM. And its cell wall is made up of a variety of different sugars and different proteins. And it's different than Cryptococcus neoformans, okay? So these are both can infect people, but their cell wall compositions are very different. And even if we look across in a single organism across developmental time, we can see differences. So these are two different growth forms of Aspergillus, and you can see that their cell walls are also quite different. So, but despite the fact that cell wall composition varies widely across dikarya, they generally include polysaccharides, okay? It's not an absolute rule, but this is a sort of a general fact. And so these polysaccharides are assembled by enzymes that are produced or that are membrane bound. And so, for example, we can think about enzymes that make chitin called chitin synthases. And so these membrane bound enzymes will take precursors from the cytoplasm and link them together to make these cell wall polysaccharides. And so this means that the localization of these synthases directs the polarized growth of cell wall and dikarya. So for example, here's a filamentous fungus. And here we're looking at a GFP tagged chitin synthase, and you can see it's localizing to the tip. And so the cell is actually depositing these chitin synthases at the tip, and you're getting more cell wall incorporation at the tip, and that's why you're growing more at the tip. So really the question becomes then, how do cells control the localization of these enzymes? And so these synthases are trafficked around dikaryotic fungal cells in vesicles. And the localization of these enzymes is controlled by the actin cytoskeleton in two ways. The first is there are structures called actin cables, which can help direct the deposition of these chitin synthases at localized sites. So here is a picture of a budding yeast. It's a mother cell is the big cell, and the little one is the daughter cell. And you can see these actin cables in the mother cell. And these actin cables, among other things, can help direct vesicles containing cell wall synthases to the bud neck, where the daughter cell is budding off. The second way that the actin cytoskeleton can control the localization of these synthases is by controlled internalization of the synthases. So endocytosis in fungi is mediated by actin structures called actin patches. And so they will direct the internalization of these synthases from sites where you no longer want to have the synthases actively producing new cell wall material. So for example, in these filamentous, these polarized growth here, you're depositing the synthases at the tip, and they're diffusing around in the membrane, and you're pulling them in from the side, okay? And so you're going to have localized concentration of these cell wall producing enzymes at the tip. Okay. And you can see these actin patches in this daughter cell. These are these bright actin puncta. And so that's driving endocytosis of different, many different things, including different types of cell wall synthesis. And so this means that fungal morphology relies indirectly on the actin cytoskeleton. And that's in contrast to what we see in the lineages surrounding fungi, which use the actin cytoskeleton directly to control the cell shape. And so together, this means at least 3 major changes must have happened during the development of fungal morphogenesis, right? First, we have the evolution and expansion of families of enzymes that are producing cell wall material. You also have differential regulation of pressure. So I told you that fungi, or dikaryotic fungi, use turgor pressure in order to inflate their cell walls and to grow. But it turns out that if you're an animal cell or an amoeba and you have high internal pressure, that's a big problem because you don't have a cell wall on the outside of the membrane. So you're literally going to explode. So there has to be a difference in the regulation of internal pressure. Finally, we have this drastic change in the actin cytoskeletal networks that help control cell shape. Okay. So how can we go about studying the development of these systems, which together give rise to fungal morphogenesis? And there are 2 sort of classic ways people have approached this. The first is using comparative genomics. So we can compare the genomes of fungi and non-fungi and identify genes that are shared or that are different. And we can also do comparative cell biology. So we can look at the structures and behaviors of these different cell types. And that can tell us a lot about what's going on here, but it can become very tricky to understand which of the differences in these organisms that diverged over 800 million years ago are really specific to the morphogenic programs and which are causing differences that are upstream or downstream. So to complement these approaches, we are interested in finding an internally controlled system that can help us compare these different morphogenic programs. And that's where chytrids come in. And that's because chytrid fungi have a biphasic life cycle that include both animal-like and dikaryotic-like or yeast-like cell types. So chytrids start their life cycle in this cell type called a zoospore. This is a cell that has a posterior flagellum that it can use to swim through the water column. And these cell types do not have cell walls, okay? No cell wall. And so in many respects, they resemble a little bit like a human sperm in that they do directed cell migration, they don't have a cell wall, and they have a posterior flagellum. They then transition to their growth form called a sporangium. And they do this by retracting their flagellum, so they're no longer motile, and they build a chitin-containing cell wall. And so, and in many respects, their growth form resembles dikaryotic fungi, like a yeast. And this property of having features that are shared with animals and other non-fungal lineages, and other features that are really reminiscent of dikarya, is a function of where chytrids sit on the tree. And so chytrids are a non-monophyletic group of fungi that branched very early in fungal evolution before the radiation of the dikarya. So they have retained many features that have been lost in dikarya. And so my lab is developing chytrid fungi as genetic systems to study the development of fungal morphogenesis. And we primarily studied two species. The first is Batrachochytrium dendrobatitus. Okay, we call it BD because that's a mouthful. And this was called by Ed Young the worst disease ever recorded because of the diversity of species that this fungus can infect and kill. So this is an ongoing ecological disaster. BD is all over the planet and has ravaged a number of different amphibian populations and driven a number of species to extinction. So we studied the basic biology of this system in the hopes that understanding something about the cell biology may be useful for people that are trying to develop methods of environmental remediation. We also study Spizellomyces punctatus. This is a non-pathogenic chytrid fungus. And we have two systems because the genetic diversity of chytrids is vast. And so we want to understand, are the things we're learning from one chytrid species likely to be true of the group as a whole, or and which are more lineage specific. So I'm going to talk more about fungal morphogenesis in a minute, but I want to give you an update on some genetic tools. And the stories I'll tell you, we're now sort of following up on a molecular level using some of these tools. So last year, we published our first paper showing transformation of BD, this frog-killing fungus. And we do this via plasmid electroporation. So we can do fun things like select for antibiotic resistance, which isn't fun in itself, but allows us to do a lot of fun things. Like express and select for cells that are expressing luciferase reporters. So we now have expression reporters, so we can look at different promoter activities from different genes across the life cycle. We've done that for a variety of promoters now. We can actually electroporate in multiple genes at the same time on different plasmids and see expression of both of them. So we can do quite a lot with a single experiment. We can now do protein tagging and a variety of other things. But the system is inherently limited for two reasons. One is the transformation is transient. So these cells take up the plasmid and express from it, but don't retain it. And secondly, we can do heterologous gene expression off of a plasmid, but we really want to get to gene knockouts as well. And so our next step was to develop homologous recombination. And we've got that working now as well. So the first thing we did was to target the GAPDH locus, that's glyceraldehyde 3-phosphate dehydrogenase, not because we're interested in that enzyme, but because it's a really common enzyme that we know that you can add things to and it still functions okay. And so we developed a gene targeting vector that had homology arms to the GAPDH locus that allowed insertion of a fluorescent protein to the C terminus of GAPDH, as well as an extra cassette for expression of hygromycin resistance protein. And so if we get correct insertion, we should get both expression of a GAPDH fluorescent protein fusion as well as hygromycin resistance. Oh, bad slide here. Okay, so we put this in and we select for hygromycin, and we can detect the modified locus using PCR, and we confirm this with whole genome sequencing. So we know that this is going into the locus that we want and nowhere else. And then we can start looking at expression of this thing. So we did Western blots using an anti-GAPDH antibody, and in our wild type, we have a ton, looks even more on this projector, of GAPDH. And then in our homologous recombination, we can see another band. We think it's the tagged locus. And we can use like an anti-RFP antibody, and we see only a band in the hygromycin resistant line. And then we can do fluorescence microscopy. So normally these things are completely transparent when we illuminate for RFP, but then when we have hygromycin resistance, we also see RFP expression. So this is, we think it's working well. And we've actually used this now to make our first mutant. So we did this by targeting the URA3 locus for a reason I'll get to in a moment. And we first put in targeted replacement of the URA3 gene with hygromycin resistance. And when we do this, we get cells that are resistant to hygromycin, but not resistant to another antibiotic, blastocidin. And then we targeted the other URA3 locus with a blastocidin resistance gene. And so when we do that, then we get cells that are resistant to both hygromycin and blastocidin. So this should be, in principle, a URA3 knockout. And the reason we chose URA3 is because a trick. There's a compound called 5-FOA, which if you expose it to cells that have functional URA3 protein, it turns it into a toxin, okay? And so you should be able then to kill any cells that have a functional URA3 copy. And so we take these cells and we expose them to 5-FOA. We get cells that are resistant to 5-FOA with our double knock-in line. So we now have done genome sequencing of all of these lines as well, and we're just waiting to confirm that we don't have a insertion of any of these things anywhere else. But the fact that they're 5-FOA resistant, I think, is really promising. So anyway, we're really excited because this means that we can now make a targeted deletion of a gene of choice using this tandem homologous recombination replacement strategy. Okay, great. So we are now developing more tools for these systems. I'm happy to talk about tool development. I think it's really a fun set of projects, but I really want to go back to talking about the basic biology of chytrids. Okay, and so we have these 3 different programs that must have changed during the early evolution of fungi. Today, I'm not going to talk about cell wall pathway evolution. I'm really going to talk about actin evolution. And then at the end, I'm going to share some unpublished data talking about differential regulation of pressure. Okay. So I want to give sort of 3 vignettes that really highlight why I think chytrids are a fantastic system to study the evolution of the actin cytoskeleton. The first is this idea that chytrids are an evolutionary Rosetta Stone that can help link our understanding of the actin cytoskeleton of yeast and other dikarya with our understanding of the actin cytoskeleton of animal cells and amoebae. Second, chytrids display a really remarkable developmental separation of actin phenotypes. And third, we found that exposure to host factors can actually trigger a switch from one developmental phenotype to another. Okay, so what do I mean by an evolutionary Rosetta Stone? Before I get there, I want to give a little bit of background on actin, because I'm guessing not everybody in the audience is obsessed with actin as I am. But actin is an amazing protein that can form filaments, but by itself, it's actually really terrible at forming filaments. And that's because to make a filament, you have to get 3 actin monomers organized in exactly the right way to make a stable seed, and then that can elongate. So this results in a kinetic barrier to polymerization. And cells use this kinetic barrier as a key regulatory step. And so there are 2 protein complexes called nucleators that help cells overcome this kinetic barrier to actin polymerization. And both of these were present, we think, at the last common eukaryotic ancestor. So these are super ancient systems to control actin regulation or actin polymerization. The first is the ARP23 complex. So this combined to the side of the existing actin polymer and see the formation of a new actin polymer. So this results in branched actin networks that are really good at producing pushing forces. Cells can also nucleate actin filaments de novo using Foreman family members. And so this results in unbranched actin networks. And so these unbranched actin filaments can associate with myosin motor proteins to make constricting forces, okay? So the same polymer system can produce pushing forces and pulling forces depending on the architecture of the network it forms. And so cells will combine these different types of networks together to give rise to a wide variety of different phenotypes. So actin does a lot in eukaryotic cells. And it turns out that these nucleators themselves are also heavily regulated. They can be regulated at the level of actin isoform expression or actin monomer binding proteins. These nucleators are also controlled by upstream factors, cleverly called nucleation promoting factors. And those are in turn controlled by a wide variety of upstream factors. So you have many layers of regulation, and this is just at the level of nucleation. And it turns out that actin turnover, polymer turnover, is also heavily regulated, as is the interaction of actin filaments with other structures. So this is all to emotionally prepare you for the next slide, which is a lot. But we built what we call a jelly bean plot. So here, each column represents a different family of actin regulators, okay? And each row is a different species. And the color of the jelly bean represents the copy number of that regulator that we find in the genome. So this is Homo sapiens, this is us. And you can see we have many copies and many actin regulators. And that's fundamentally different from what we see in dikarya. So dikarya are missing many of these actin regulators altogether and have many other in single copy number. When we look in chytrids, we find they have an intermediate complement of these. So they have actin regulators that are missing from dikarya, as well as others that are found in higher copy number. And I'll talk about two today. I won't talk about them all. That'd be fun, but it'd be a long time. But I'll give you two examples of the type of pattern we see. The first is the SCAR complex. So this is one of those activators of the ARP23 complex. And we find it in animals and other non-fungal lineages. And we find it in many chytrid species, but never in dikarya. And this complex is really interesting to people that study cell migration because it's really important for driving cell crawling. So cell crawling is a mode of movement that relies on actin that's really common in animal cells as well as amoebae. And these cells will crawl using these structures here. They're called pseudopods, okay? And they're filled with actin. And this actin is templated by the SCAR complex. So we have localization of the SCAR complex at the tip of these structures, which is going to seed the formation of these new actin branches. So you're going to get more and more actin growing at the tip, and so the structure is going to grow. So the fact that we saw the SCAR complex in these chytrid species, but not in dikarya, led me to wonder, can chytrids actually crawl across slides like an amoeba or a white blood cell? And so I spent the back half of my postdoc sort of thinking about this and exploring this idea. And I really focused my attention on the zoospore form, because that's the cell type that doesn't have a cell wall. And you can imagine how having like a stiff fungal cell wall would make it difficult to make these dynamic structures. And to cut, maybe a couple years of work into one video, it turns out that chytrids, well over 80% of them, of the newborn chytrids, crawl and build these actin-filled pseudopods. So this is a chytrid zoospore. It still has a flagellum, okay? And you see it twitching a little bit, but it's not beating, okay? So the movement that you're seeing here is being driven by the structures at the front of the cell, not the flagellum. And so these cells will crawl around 20 microns per minute. That's similar to a white blood cell or many types of amoebae. These are cell types that people consider to be professionally migratory. So this is like a really robust migration. And these structures are filled with actin. So here we're staining cells with phalloidin, which is a small molecule which binds to polymerized actin. And you can see it enriched in all of these structures. So we can also explore the reliance of actin polymerization for these structures using small molecule inhibitors. So because actin does so much, an actin mutant isn't particularly informative because the cells are really unhappy for a ton of different reasons. And for that reason, the cell biology community has really honed the use of drugs that allow us to interrupt actin cytoskeleton at precise time. And so one of these is a drug called latrunculin B. It binds actin monomers and makes them unavailable, soaks them up. And so we no longer see polymerization of actin. So when we treat these zoospores with latrunculin, we see that they stop making these structures. And if we wash it out, they start making them again. So this means that these structures are reliant on active actin polymerization. We can also treat cells with another compound called CK666. This is an inhibitor of the ARP23 complex. And we see the same pattern. And so we believe that these structures are really relying on actin polymerization, but really this branch actin polymerization, just like white blood cells and amoeba do. So now we can start using some of our genetic tools to look at this in living cells. So here's a movie of another crawling BD zoospore, and DIC is on the left, and we're expressing LifAct, which is a 17 amino acid peptide that binds to polymerized actin, and we fuse it to our red fluorescent protein. So we can see in living cells where actin polymers are, and you can see that they're, again, these are dynamic networks that are being built at the leading edge of the cell. Okay, so this means that the presence of these animal-like actin regulators is able to predict cell crawling in these lineages, which is, I think, pretty amazing. The other example I want to talk about are the Formin family proteins. These are those nucleators that make these unbranched actin networks. And you can see that animals and chytrids have many copies of these, and dikarya, some species have many and some have few. But it turns out that formins have undergone a huge amount of evolution as a family since the last common eukaryotic ancestor. So to understand more about the subfamilies of these formins, we built a big phylogeny. You're not supposed to be able to read the leaves of these, don't worry about that. But we've colored the genes based on the lineage from which they come, that the organism is in. So you can see animals are in blue, dikarya in orange, and chytrids and their sister group, Blastocladiomycota, are different colors of pink. And so you can see that there's color spread all around the tree. It looks like, you know, some sort of circus. And this is indicative of early diversification of this group of proteins. So it's not just that the last common ancestor had formins, the last common eukaryotic ancestor had many different types of formins. And so I want to talk just about two subfamilies. The first are these formins. They're called dam formins. These are important in animal development. They help link the actin cytoskeleton to microtubules. And we find them in every species of chytrid we've looked at so far, but never in dikarya. Okay, so this is an example of a formin subfamily that we think was present at the last common ancestor of fungi that have been retained in chytrids, but lost in dikarya. The second example I want to talk about are the BNI1 formins. These are formins that we find in every single dikaryotic genome we've looked at so far. So they're ubiquitous across dikarya as far as we can tell. And we see it in some chytrids, but not all, but we don't see it outside of the fungal kingdom. So this is an example of an actin regulator that seems to be unique to the fungal lineage that's been lost in some species, but seems to be sort of uniting them. So the fact that chytrids have actin regulators that they share with animals that we don't see in dikarya, as well as other actin regulators that they share with dikarya that we don't see outside the fungal kingdom, is why we think of them as a Rosetta Stone. Because as cell biologists, we understand a lot about the actin cytoskeleton of yeast. We understand a lot about the actin cytoskeleton of animals, but how those two lineages diverge from each other is kind of a mystery. And here we have a system that can help us sort of understand that. Okay, so next I want to talk about what chytrids are actually doing with all these different actin regulators and talk a little bit about their development. So as I mentioned, chytrids have two main cell types. They have the zoospore and the sporangium. So we first looked at all the different actin structures that the zoospore can build. And I already showed you that they build this actin-filled pseudopods when they crawl. They also build these structures that we're calling spikes. These are linear structures. They visibly resemble phylopodia on animal cells, and these are finger-like projections animal cells use to sense their environment. We have no idea what they're doing in chytrids. And they also have an actin cortex, okay? And this is the structure, this is this actin layer under the membrane that they're using to control their cell shape when they don't have a cell wall. And so this is fundamentally different from what we see in sporangia. So these are 3 BD sporangia. The round part is the cell body. And you can see these filamentous structures. These are called rhizoids. These are structures that the cell uses to anchor to a substrate and can increase the surface area at which these cells can take up nutrients from the environment. When we stain these cells for actin, we can see these really bright actin puncta that visibly resemble the actin patches that we see in yeast and other dikarya. So the actin patches in yeast and other dikarya are peripheral, so they're on the surface. They're dynamic, they turn over pretty quickly, and they're ARP2/3 dependent. And we see the same thing here. So we really think that these structures are likely driving endocytosis in chytrids like they do in dikarya, but we haven't formally tested that yet. So this suggests that maybe these sporangia might be more similar to dikarya, but there's something that dikarya have, which are these actin cables. And we didn't see that in these cells in BD. But it turns out that these actin cables are built by BNI1 formans in dikarya. And I told you that BNI1 formans are found in some chytrid species, but not all. And it turns out that BD is one of those examples that doesn't have BNI1 formans. But luckily for us, our other model systems, the Zellomyces does have BNI1 formans. And when we look at the actin cytoskeleton there, we can see, again, these peripheral actin patches, as well as these really bright actin cables on the inside. So this suggests that maybe these BNI1 formans in chytrids might also be making actin cables like they do in dikarya. And so we did this before we had genetic tools. So we thought maybe one way to test this is to see if the presence of or absence of BNI1 formans could predict the presence or absence of cables. So we did that experiment. I already showed you that BD doesn't have BNI1 formans and has no cables. SP has these formans and makes cables. We looked in BSAL, which is another chytrid that doesn't have BNI1 formans, and we find that they don't make cables. And then we looked in RG, which is another chytrid that has BNI1 formans, and we find that they do make cables. So it's a correlation, but it's a correlation that can actually predict the phenotype. Okay, and so coming back to this question about what chytrids do with all these different actin regulators, we now understand that the chytrid zoospore uses actin to be able to crawl and has built an actin cortex and these actin-rich pseudopods, while the sporangia builds actin patches and in some species at least builds these actin cables. And so the fact that these chytrid zoospores could crawl raised a really obvious hypothesis that maybe they're using these genes that in animal homologs drive crawling motility in order to achieve this phenotype. And so to test this idea, we figured out how to synchronize the development of these organisms and then did RNA-seq at different stages. And what we found is that these cells specifically express these crawling genes in the zoospore cell type, while the sporangium, the growth form, expresses homologs of genes in yeast that drive endocytosis. And so really we're thinking about this as these zoospores of having animal or amoeba-like actin cytoskeleton in terms of both the structures they build and the actin regulators they express, whereas the sporangia are yeast-like in terms of the structures they build and the genes they express. And this data and other data I don't have time to go into has really led us to think that the dikaryotic fungi evolved from an ancestor that had animal-like actin regulators and animal-like phenotypes. And this means that the simplicity we see in the actin networks of yeast and other dikarya today is not ancestral, but is a secondarily derived phenotype, maybe that evolved due to gene loss in the fungal lineage. Okay, great. And so now that we know that chytrids display this developmental separation of actin phenotypes, we were really interested in understanding whether some of this actin biology might have some import on the ecological role of BD in the environment. Okay, and really we started thinking about this in a really simple way. And we know that this process of transition from the zoospore to the sporangium is a really important moment in a chytrid's life. It's a unidirectional transition. So once they undergo this process we call encystation, the transition from the zoospore to the sporangium, they don't go back. They have to go all the way through the life cycle before you'll see more zoospores. And we see drastic changes in the actin cytoskeleton, which I've shown you. The flagellum also gets internalized, so they're no longer motile. And they transition from not having a cell wall to having this chitin-containing cell wall. So we wondered, how does BD decide when to undergo encystation? And we did, again, some very sort of kindergarten-style thinking. We thought, well, the zoospore is probably about finding the host, and the sporangium is definitely about growing on the host. Maybe arrival at the host might be a good time to undergo this encystation reaction. And I told you that BD grows on frogs, and it turns out that BD grows in the skin of frogs and other amphibians. And you might think about the skin of frogs as being slimy or gloopy, and that's because frogs and other amphibians, their skin is coated in mucus. They have this mucosal layer. So we thought, well, maybe BD might be able to sense its home on a frog by exposure to mucus. So we partnered with the local USDA Fish and Wildlife Station and got mucus from local wild green frogs, which can be infected with BD. And so on the left are cells that are just exposed to buffer. And on the right are cells that are exposed with buffer mixed 50-50 with this mucus from these wild green frogs. And you can see within minutes, these cells round up and retract their flagella. These are key signatures of encystation. So we thought, wow, it's a really rapid, reproducible, and synchronous response. We thought, what in mucus might be doing this? It turns out that amphibian mucus is a really complex mixture. The bulk component is mucin. These are these heavily glycosylated proteins, and so it makes mucus gloopy. But also in amphibian mucus are a wide variety of different microbes that live there, as well as these small bioactive peptides. A lot of them are antimicrobial. So, at first we're like, we'll never figure out what in there is doing it. But we decided to do the easy experiment first, which is just to test purified mucin. And it turns out that purified mucin is sufficient to trigger encystation. So here you're looking at the percent of cells that have undergone encystation with various concentrations of mucin. This is the percent that have undergone encystation in 5 minutes, okay? So we can see with, 1 mg per mil, about half the cells undergo encystation. And we sort of hit peak response at around 10 mg per mil, which is a similar mucin concentration that you see on the skin of fish. So we think it's kind of in the realm of physiological concentrations. And we think this really is encystation because we can stain the cells and look at the internal structures. So the top row are cells that were treated with buffer for 5 minutes. On the bottom are cells that were treated with 10 mg per mil of mucin for 5 minutes. And you can see that the mucin exposure causes the flagella to be retracted inside the cell. The actin cytoskeleton has switched from pseudopods and cortex to patches. And we go from having no detectable cell wall to having a readily detectable cell wall via a calcium white stain, which is a common cell wall stain. So this is a very fast reaction. We're talking 5 minutes. So, as a cell biologist, I wonder, how are these events coordinated? And the first, obvious idea is that maybe there's gene expression changes, but we can treat these cells with tons of cyclohexamide, which blocks protein synthesis, and they still undergo the reaction just fine. And this is consistent with old literature from other zoospore fungi, which also don't require protein synthesis to undergo this developmental change. Also, 5 minutes seems awfully fast for this process to be controlled by transcription and translation. So we don't think it's gene expression. And then the other obvious idea is that there's some sort of second messenger cascade. And really, we were inspired by what happens in animals after fertilization of the egg. And so the sperm will come fertilize the egg, and there's this huge cascade of events that happens. And these events are coordinated by a big calcium flux. So we thought, let's check calcium. And so based on these experiments I'm about to show you, we think that calcium flux is both necessary and sufficient to induce encystation. So the first set of experiments we did was to test the requirement for calcium in encystation. So we treated cells with BAPTA. This is a calcium chelator. You can think about like a sponge to soak up calcium. And so when we treat cells with BAPTA, they no longer undergo encystation, even if we give them mucin. So calcium is required. We next treated cells with ionomycin. This is a calcium ionophore that can carry calcium across membranes. And so if we treat cells with ionomycin, even in the absence of mucin, they rapidly undergo encystation. So it seems like a pulse of calcium is sufficient to induce encystation even in the absence of mucin. So this is the data that we have that shows us that we think calcium is involved in this signaling response. So our working model based on these and other data is that BD is using mucin as a host cue to induce the switch from this dispersal phase to its growth phase. And the way we think that's happening is that BD zoospores have their cell wall enzymes ready to go in vesicles, such that when they get exposed to mucin, these vesicles can then dock at the membrane and start producing cell wall material right away. And so this is a simple way in which BD can coordinate its life cycle with arrival on the host. So here we have this dispersal form, right? And if you don't move to your growth form, you're likely to swim away from your host, which is not good for you. But if you sense mucin and you induce this switch to the sporangium, then you sort of coordinated your life cycle with that of your host. And so this model really raises other interesting questions, like what if a species evolved to sense something other than mucin? It would then be able to coordinate its life cycle with other things. And I think this is a really interesting idea because it turns out that there's over 1,000 chytrid species, and the genetic diversity is as great as all of animals. So it's a hugely diverse group of fungi. And members of this group form really important symbioses with a wide variety of organisms, from plants, animals, other fungi, you name it, algae, whatever. And these symbioses can range from parasitism like BD to mutualism. And so we're exploring this idea that maybe this developmental switch could be hijacked by different signaling cascades to allow these organisms to quickly evolve to coordinate their developmental cycle with different host species. Okay, so anyway, these are the reasons why we're so excited to use chytrids to study the evolution of actin in the episticont lineage, because they have these phenotypes that resemble at times animal cell phenotypes and other times dikarya. They have actin regulators that they're sharing both species, and now we can, or both groups, and now we can control the switch between these different cell morphs. Okay, so I want to spend my last remaining minutes talking about what we know about the regulation of pressure in the system. So we know that chytrids use cell wall enzymes to make their cell walls, but dikarya uses both cell wall synthesis as well as turgor pressure in order to grow. So we first asked whether chytrid sporangia also need pressure in order to grow. And in dikarya, internal pressure called turgor pressure is related, controlled by controlling water flow in and out of the cell. So for example, we have a normal yeast cell, and if we give it a hyperosmotic shock where we lower the water concentration outside the cell, water will leave the cell and the cell will shrink a little bit. And the cell senses that and upregulates synthesis of glycerol, which then lowers the concentration of water on the inside of the cell, and then water will flow back in and the cell will puff back up, okay? And so you can test for turgor pressure by changing the external osmotic environment. And then you can check for control of turgor pressure by seeing how cells respond to that change in the osmotic environment. So we asked first what happens then when we hyperosmotically shock chytrid sporangia. So on the left are cells that are treated with 0 millimolar sorbitol, and on the right are cells that are treated with 300 millimolar sorbitol. Sorbitol is a sugar, it's a non-metabolizable sugar, and it's a very simple way of controlling external osmolarity. And so we're looking at the cell wall. And so you can see that these cells, when they're treated with sorbitol, they're shrinking a little bit, okay? And we can then treat cells with 500 millimolar sorbitol, and these cells shrink a lot, okay? So these cells are definitely under pressure, and we know that now because if we change the external osmolarity, that these cells will shrink. But that doesn't tell us that these cells are actively controlling internal pressure. To do that, we need to really look over a long time and see if the cells can actually recover their volume. And so here I'm going to show you an hour-and-a-half time lapse of the same experiment. And you can see that these cells quickly shrink and then they start to reinflate, okay? And so this is diagnostic of regulated internal turgor pressure. Okay, so we now know that these cells then are able to regulate internal turgor pressure, have high pressure. What about the zoospores? And because they don't have a cell wall, so they can't maintain high turgor pressure because then they're going to explode. And this is actually an even bigger problem for chytrids because they live in freshwater environments. And so freshwater has very low solute. And so organisms that live in freshwater are continuously battling this influx of water. And so other freshwater organisms have specialized organelles that help them deal with this. And so these organelles are called contractile vacuoles. This is Naegleria amoeba, and it has this structure in the back. You can see this vacuole that's pumping, okay? And so these vacuoles act like they're bailing water out of a leaky boat. So they'll collect water and then they'll literally pump it out of the cell. And organisms from across the eukaryotic tree that live in freshwater that don't have cell walls have these structures called contractile vacuoles. And so my lab accidentally started working on contractile vacuoles. That's a story I can tell you later if you want. And we've been studying them in this amoeba Naegleria. So we were primed to ask, do chytrid zoospores then also use contractile vacuoles? And so these contractile vacuoles come in a range of different morphologies, but they all work on the same underlying principle. And that's by using vacuolar ATPases to make a gradient to allow water to flow into the vacuole. And then these vacuoles open to the outside of the cell, and the internal pressure inside the cell pushes on the vacuole to push the water out of the cell, okay? It's a literal pump. And so we started imaging chytrid zoospores. These cells are very small. They're around, 2 microns in diameter. But we see these vacuoles here. You can see some here that are coming and going. They grow and then they quickly shrink. So those looked like they could be contractile vacuoles. But the diagnostic test of contractile vacuoles is that their activity depends on the external osmolarity, right? Because that's what's driving water into the cell at different speeds. So here's a cell in 0 millimolar sorbitol. You can see it has a contractile vacuole that grows and then quickly shrinks. And these are cells that are in 100 millimolar sorbitol. We can see no obvious vesicles that look similar to these, what we think are contractile vacuoles. And so we got really quantitative about this. We measured the size of each of these things under different external osmolarities, and we find that they disappear at 100 millimolar. We looked, and instead of the total of all of these vacuoles, we did the biggest one, and those also go away. We measured the disappearance rates, and those just bottom out as well. And so we can multiply these things together to get an idea of the amount of volume that is changing due to these activities under different sorbitol concentrations. And we see the activity goes away. So this is the diagnosis of contractile vacuole. So this means that chytrid zoospores are actively maintaining low pressure, while the sporangia are actively maintaining high pressure. And then this is coupling with this idea that we have these animal-like actin networks in the zoospores, and we have dikaryotic-like actin networks in the sporangia. And so this is the reason why we're really interested in using this as a system to study animal-like morphogenesis and fungal morphogenesis. And so together, this means that chytrid fungi are an internally controlled system to study the evolution and development of fungal morphogenesis. And now that we can control this in the lab, the transition between a zoospore and a sporangium, it's a really amazing tool set. So here's a cell that's undergoing encystation, and we're looking at the actin cytoskeleton. So we can combine our genetic systems with the ability to control the rapid transition from this animal-like cell type to this yeast-like cell type. And so we can complement these experiments that comparative genomics and comparative cell biology, where we're looking at cell types that diverged, over 800 million years ago, and complement that work with experiments in the lab where we have one organism with one genome. We're comparing the biology of these 2 different cell types that are separated by minutes of development. Okay, so with that, I got lots of time for questions, but I want to thank my lab members. I want to especially thank Edgar Medina. He's in the top right. He has been really hard at work developing agrobacterium-mediated transformation for Spizellomyces. And I want to thank Sarah Prostak. She's right here. I did put out my pointer in the white shirt. She started in my lab as an undergraduate and did a lot of that comparative genomics during her undergraduate work, and is now a graduate student who's working on this change in pressure in the system. It's been really great. So, and I'd like to thank our funders, and I'm happy to take any questions. Thanks. Yeah. Okay. So we have tried to quantify. So there's the structures. So there's the flagellar absorption, the retraction, the actin cytoskeleton switch, and the cell wall assembly. Then there's the switch from the contractile vacuole activity to Turgor pressure. And then there's like, I guess the cell wall synthesis I already talked about. So we cannot uncouple temporally the retraction of the flagella, the switch in the actin cytoskeleton, and the cell wall assembly. That's all happening really fast. The reason we haven't been able to do that is those experiments we did using fixed cells, and the timing was so on top of each other that in bulk we couldn't see a difference. Now that we have markers in live cells, we could probably sort of start piecing that out. What we do know is that they start building the wall, and they build the wall for about a half an hour before the contractile vacuole activity slows down and stops. So they're building the cell wall for a while, which I think kind of makes sense. They need to make one with enough integrity to withstand the force that's coming from the increased water flow into the cell if you're not pumping that out. Yeah. 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 Yeah, that's a great question. So two things. One is we don't actually know really the cell wall composition in BD. So Don Shepard at McGill is sort of exploring that. We can also stain cell wall components using WGA, wheat germ agglutinin. And when we do that, it stains different structures, and those structures also change over developmental time. So we do know that the cell wall composition is changing over developmental time, but in what ways exactly, we don't know yet. Yeah. Yeah. Yes. Yeah, we don't know in the wild what they're doing, but in the lab, BD can remain a zoospore for about 16 hours. If in the lab setting, they will undergo incestation just fine without mucin. So mucin can trigger it, but it's not required for incestation. I have a hypothesis for which we have no data, but I think it's probably true. But so zoospores are non-feeding, and they have a lipid droplet that they use as a carbon source. So my hypothesis is in culture that they remain zoospores until that droplet gets depleted, and then it's like, do or die, let's go, and they undergo incestation. That's an easily testable hypothesis, but we just haven't done that yet. Yeah. Yeah. Yes. And so they also are crawlers. There's also chytrid species that have flagella that do not crawl. They also don't have the scar complex or talon, so that's also consistent. Yeah. Yeah, so yeah. So the mucin data I showed you is using just the commercially available purified mucins, right? So we worked very hard to get that frog mucus, and there was no way we would get enough to purify the mucins from there. We tried, sugar moieties that make up the sugar chains on mucins. Those independently don't trigger. We tried methylcellulose, which has some biophysical properties similar to mucins. That doesn't trigger it. We tried different proteins and amino acids. Doesn't trigger it. So there seems to be something in the frog mucus, and we think also in these purified mucins that we can't recapitulate with these single components. Whether there's some byproduct that comes with the purification that might be the thing, I can't rule that out. Yeah. Yeah. Yeah, so we know that calcium is required. And we know that we can, if we pulse cells with calcium, they'll undergo incystation. That's the data that we have. Calcium as a second messenger really doesn't work alone. And so we've been looking through lots of different inhibitors. And we use broad spectrum kinase inhibitors, we get the same effect. So we suspect that there might be a kinase involved too in the pathway, but we don't know for sure. We don't know what it is. Yeah. Great. Thank you.